Well, human biology class, this may be the last lecture. I'm not sure if I'll get through everything and need to include a short third one or additional one on the nervous system. My intention is to get through the remainder of the material I want to get through today, and then also post a video that I want you to watch as sort of a culmination to what we'll be some of what we'll be talking about today. So look for that on Moodle. You will have to, I believe, access it through Drive, the video. It will not be a YouTube video. So anyway, just if you click the link, you should be able to get there if you're on Moodle to the video. Okay, thank you. So here we go. Last time we were talking about cells that support your nervous system. We looked at this page here. We talked about those that form a protective wrapping around neurons, specifically axons, the part of a neuron called an axon that helps protect it from essentially short-circuiting. Others that form a blood-brain barrier to keep, they cover the capillaries in the central nervous system, brain in particular, in an attempt to keep, additionally keep things out of the brain that it doesn't want and only let in just those things that it does want in. There are ependymal cells that produce cerebral spinal fluid, microglial cells that kind of act like a specialized white blood cell wandering around looking for pathogens. And so there are all these support cells that I talked about some of last time. I talked about oligodendrocytes and Schwann cells and astrocytes mostly. So if you look in your notes then, right here, we're looking at then, or shifting now to the function of neurons. But before we do that, this line here, it's important because I mentioned most neuron cell bodies are in the central nervous system. That's the brain and the spinal cord. The exception would be those found right here in these little ganglia that are right next to the spinal cord but are not technically attached to they're attached to, but they're not part of the spinal cord. And you can see, so there are some neuron cell bodies in these ganglia right here in your spinal column next to your spinal cord. But there and brain and spinal cord are the only places you find neuron cell bodies. It is axons and dendrites that extend out from those areas to the rest of your body and allow you to sense your internal and external environment and allow you to react to it. So, yeah, wanted to make that point. So then if we look here, we're going to shift gears to how neurons work. And the first line here is neurons are a polar structure. And I wanted to show you a clip from a video that is linked on Moodle to make that case. So here would be a membrane that would surround, say, an axon. So here would be the inside of the neuron. This would be the outside of the neuron. It says extracellular, outside of. And so this would be inside. And so this would be the membrane that lines a part of a neuron. We're just calling it an axon. It could be any part of a neuron. Okay? And the point here is that the outside has lots more pluses on it. And that's because there's lots of these green sodiums and potassiums out here. And the inside also has sodiums and potassiums, but it also has these minuses. And the video clip that I'm going to have you look at, it is very jargony, folks. So you want to just keep your mind on the big picture items like membranes are more positive on the outside, more negative on the inside. The reason they're more negative on the inside is because there are also large protein molecules in here that lend a net negative charge to the inside of the neuron cell membrane. Okay, so that's left out most times. Bottom line is positive outside, negative inside, and it's the arrangement of the sodiums and potassiums that we want to pay attention to. So if you look at your notes, there's a couple of steps there, basically 3 steps. Number 1, a stimulation comes along. And the video that you see here has a good description of summation. Your body is constantly sensing its environment and ignoring a lot of information that doesn't rise to the level of needing to be responded to. But when information is sufficient that it needs to be responded to, Typically that's because your body is receiving lots of stimulus. That stimulus will sum up, if you will, at a neuron, and if it crosses a threshold, it'll create what's called an action potential, sort of a wave of activity that comes down the neuron and transmits a signal, an impulse that something is happening around you, or that you need to do something about what's happening around you. It'll carry that message down this membrane. So number one, there is stimulation. It's sufficient enough to create a stimulation, whatever this information is. Sodium gates open. So the sodium gates, green right here, they will come wide open, and the sodium that's outside will flush in. And when that happens, the inside of the membrane becomes a positive. It basically depolarizes, they say, because the inside becomes as positive as the outside. And so if we look here, see if I can find where that's going to happen. Yeah. Here, the sodium gates are opening. Sodium rushes in, and now the inside is positive, and the outside, well, they show negative. That's a little bit of a misrepresentation, but basically you can see there's a charge difference that happens here. A depolarization. When it says depolarize, that means there's not a polarity difference inside and out. That's why I mean a little bit of a misrepresentation. It should be plus on the outside, plus on the inside, depolarized. To be polar would be what you see here, where you have minuses on the outside, pluses on the inside. To depolarize means you have pluses on both inside and outside. Okay, so then the step number 2 comes along. That's the potassium gates open. That's these purplish colored things here, because the potassiums are purple colored balls in this particular case. Okay, and the potassium gates open, and the potassium rushes out, and that restores a charge difference, so that all the potassium's out here. Now you have a positive charge on the outside again and a negative charge on the inside. The only problem is this isn't the resting state. In the resting state, if you remember, there were sodiums and potassiums about equally distributed inside and out, so we need to get some of those molecules back to where they were. And that's what happens in the refractory period. The sodium gates can open during this resting or refractory period, step number 3. So these can open, so no more sodiums can get out, and sodium is then pumped back out, and potassium is pumped back in, and the resting potential is restored. Okay, and let's see, I think I've got just a little bit of an animation here. Maybe I'll just play this here, and you can see that.
Against their concentration gradients, re-establishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly.
So hopefully you saw that. If we can go back to that. This is kind of it in a nutshell. So here's the resting state, positive outside, negative inside. The potential comes along, the stimulus comes along. These sodium channels open up, sodium rushes in. Just a brief moment later, the potassium channels open up, potassium goes out. That's step number 2, repolarization. And then that previous situation is restored in the refractory period. So let's watch that again.
As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly.
Okay, so that's all happening. It takes a lot of time to say that, obviously, but it's happening very quickly. And in reality, any impulse can move down an axon or a dendrite or over a neuron cell body very quickly. This whole thing can happen even quicker because if you remember, there are nodes of Ranvier or Ranvier. That's these nodes right here. If we zoom in on this a little bit, maybe a little bit more, and move this up. Okay, so this is again your prototypical motor neuron. Here are Schwann cells wrapped around the axon, and this is a node of Ranvier. Okay, and there are ion gates concentrated near these nodes of Ranvier. So sodium and potassium can move much more quickly near these. So what happens is an impulse will bounce, kind of like Tigger, boom, 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 from node to node to node, and can go down that axon much more rapidly than if it were not wrapped by those Schwann cells. Okay, the bottom line is this whole process, even though it took me a lot of words to say it, can happen very quickly. I'm talking 450 to 500 miles an hour. To give you an idea what that's like, well, check this out. That'd be like that, okay? That's how fast that neuron can actually even faster than that, can go down that, that membrane, okay? Now, there's a couple of, I think this kind of interesting things. If we consider this whole process of sodium, well, I'll back it up just a little bit more. These sodium channels opening when an impulse here. So here's the impulse coming along. Boom, first thing that happens, these sodium gates open up, sodium pops in. You probably all heard of puffer fish. Here's a puffer fish resting. Here's a puffer fish popped out. If you poke a puffer fish, you get poked by a puffer fish, you know, it's a bad deal because in many of them there's toxin in these spines. The way that puffer fish toxin actually works is by preventing these sodium channels from opening up. So that impulse would come along. If that pufferfish toxin gets in the way right here, boom, that impulse stops right here, and you're paralyzed. What else did I want to say? Oh yeah, Novocaine. Whoa, what a fine thought. If you've gone to the dentist lately, Novocaine decreases the movement of sodium ions into the cell. So Novocaine is going to slow this down, and so that pain stimulus that you get from the dentist drilling a hole in your tooth, it doesn't get to your brain as much and sufficiently enough for you to notice it, and that's because it slows down this sodium movement right there. Okay, so I want you to look at that video, and hopefully you can get more from it. Again, there's a lot of verbiage with it, but kind of keep your mind on your eye on the main points of it, and I think you'll do fine. If you think about sensory areas of the body, this is a guy called homunculus. There are parts of your body that are more sensitive and parts that are less sensitive. And so what somebody did is they made a clay model of the human body, but they made the parts of the human body the size that would indicate the number of sensory organs that are in that part of the body. So the more sensitive it is, the bigger it is. And so you can see hands and lips, ears, those are the biggest, most sensitive parts of the body. And if you want to look it up, it's called homunculus. If you look at your notes then, just to see where we're at here. So we've talked about this process right here. We're going to go to synapse right now. And that's very important. That's where the video kind of ends up talking about. But we've already talked about it some. We're going to talk about it in more detail now. So right here is the end of a neuron. And so if you look at another image from your textbook, I think this is a really good image. This is an image of all the synapses that are on a neuron cell body. So these would be coming from other neurons, not just one, many. But this is what it would look like in actuality. And here's an artist's rendition of that. So it's not just one neuron connecting to another neuron via a synapse, but multiple interconnections. So this is, I think, a good image to have in your mind when we talk about what goes on at a synapse. So you take one of those connections. So again, if I go back to that, if we just take one of these connections right here, one of these little blue spaces there, and we'll look at that in more detail. Right here's what we have. We have the axon turn, the end of the axon right here, and this is, it says the receiving neuron. It could also be a muscle, it could be a gland, an organ in your body, who knows, but whatever is receiving that stimulus is right here. Between the two is the cleft. Okay, the space between them. And that stimulus that's coming down this axon has to be transmitted across that cleft. And the thing that does that is something called a neurotransmitter. They're released on one side of the cleft, and they bind to receptors on the other side of the cleft. Okay, so if I move this up a little bit, here's that cleft, and we'll look at it even more closely. Here's the cleft. So there's a packet of neurotransmitters here that dumps the neurotransmitters into the cleft. They travel across the cleft and are received by receptors on the other side. Interestingly enough, if you remember, it's calcium that is required to move these little packets to the membrane, to the edge of the axon. This packet actually merges with that membrane, opens up, and dumps the neurotransmitters into the cleft. Okay? So those neurotransmitters then can't stay there forever. You don't want them to continually be stimulating this other side of the cleft. And so well, what do? Well, your body has to get them out of there. They can be either enzymatically broken down or reabsorbed and reused. Okay? So keep that whole process in mind. It's the neurotransmitters that are in the cleft. How many of them are there? How much of them are there? How long are they there? That's a big deal, folks. Okay? Another big deal is you get the impression that a stimulus coming down this axon it's just that it's going to stimulate something to happen right here on the other side of the cleft. And this is where we're going to get just a little deep in the weeds. Neurotransmitters may excite or inhibit a receptor. You have to have neurotransmitters both causing something to happen and stopping something from happening. I'll explain a little bit more when we get into it just a little bit more. Okay? Okay, here we go. Hang on. So, neurotransmitters can either cause something to happen or cause something to stop happening. These pathways, like you see right here, from this interneuron to what looks like another interneuron or a motor neuron in this case. Okay, that would be considered a pathway. Some of those pathways may excite something. They may cause you to do something, cause your muscle to do something. They may also inhibit you from doing something maybe that you shouldn't do or don't want to do. So these pathways can be excitatory or inhibitory. Good example is your muscles. Your muscles need to work when you want them to work. And so they will receive a stimulus to do something when you need to respond to your environment. Interestingly enough, your muscles also need to be told not to work. And so there would be another pathway along which a stimulus might come that would tell your muscles not to work. If you've ever had a nervous twitch that you can't control, if you've seen somebody with a seizure, well, a Parkinson's patient will have a tremor, and that can't be controlled because that inhibitory pathway isn't working completely or sufficiently. So again, there are neurotransmitters that will excite or inhibit something. There are also pathways that will excite or inhibit. So here's one for you. If you've got an inhibitory pathway and you excite it, if you send a stimulus down that inhibitory pathway, you can cause, well, you're going to cause something to not happen. I guess I'll talk about some specific examples other than get too confusing right here. Okay, so what we're going to do lastly then is look at some neurotransmitters, some specific examples. And we've already talked about one, and that is acetylcholine being the neurotransmitter that carries a message from a neuron to a muscle to do something. So we've talked about that. In lab, with the blood lab, some of you ingested some nicotine. And anybody that smokes cigarettes or is around cigarette smoke ingests nicotine. This actually mimics acetylcholine. It increases heart rate and blood pressure. We verified that in lab. And basically the difference is because you're taking into your body something that your body's not used to having, there's not an enzyme to remove it from the cleft. And so it stays in and stays active. You get a prolonged effect from nicotine use. And that's one of the issues with the cardiac situation. It stays stimulated and strains and stresses the heart. Norepinephrine or noradrenaline is the second one in the list. It excites smooth muscle and is involved with waking, dreaming, mood regulation, that kind of thing. I'm just touching the surface of these, folks, the main points. If this is of interest to you, we have a neurobiology program that goes into great depth, and it is fascinating what we know and what we don't know about this. Another excitatory, actually the last excitatory neurotransmitter that I'm going to talk about is glutamate. Now, folks, you got to realize, when I first started teaching this class, there were 25 known neurotransmitters. The first edition of your textbook, I think it's actually the third edition of your textbook, lists them. There are now hundreds that are known, and associated molecules would expand that even further. So I'm truly just touching the surface, and our knowledge of them is expanding greatly. Glutamate is the last excitatory neurotransmitter I'm going to talk about. It is the most common excitatory neurotransmitter as far as numbers of synapses that utilize it. It's involved with learning and memory. That's important because we're going to pick on alcohol quite a bit today. Alcohol actually blocks glutamate receptors. I'm picking on alcohol because I think it's been misrepresented. I actually had a post from another Bethel faculty member a few years back when Bethel was contemplating allowing faculty to imbibe, to drink. I was opposed to that, and one of the other faculty members actually said, it's time we're allowed to partake of this healthful beverage. I thought, what? Well, it's true that in some red wines you can find aspects of it that help reduce cardiac disease. Well, you can get the same thing from grape juice or from grapeseed extract. The alcohol in it, in any wine, beer, hard liquor, whatever, it's ethanol. And ethanol is toxic. It is arguably the number one health risk in the nation today. Of patients that come into my wife's hospital, well, my wife doesn't own it, the hospital where my wife works, alcohol is either directly or contributing to most of the conditions that she sees. The majority, I should say that there's nothing that comes close to alcohol. Alcohol is a carcinogen. Well, I shouldn't say that, because promoters will say it's not. Well, here's the truth of the matter. When you consume alcohol, it goes to your liver. Your liver will break it down. It will kill some liver cells. Your liver will break it down. The first thing it breaks it down into is acetaldehyde. Acetaldehyde is a potent carcinogen. So it is a cancer causer. So one of the things that it does is blocks glutamate, and it inhibits, therefore, learning and memory. Okay, we're going to continue to pick on alcohol and others. So we're going to shift gears to usually inhibitory neurotransmitters. If you inhibit an excitatory pathway, you'll stop something from happening. Okay, GABA, gamma aminobutyric acid, is, well, it's a very common inhibitory neurotransmitter. It's the thing that's in low supply in epileptics. And if you can think of an epileptic seizure, that's just this uncontrolled response to, well, I don't know if it's a stimuli per se, but just an uncontrolled reaction in the body. Valium, if you can think of Valium, diazepam, Valium, it's a calming medication for people that are too wound up. It enhances GABA. Okay, that's how it does its thing. That's how it calms you down, by enhancing GABA, an inhibitory neurotransmitter. Alcohol is a depressant, and so one of the ways that it does its depressing thing, we've already talked about, is by blocking glutamate, which is excitatory. Well, it also enhances GABA. And specifically what alcohol does is lowers behavioral inhibitions. You might say, well, that's a good thing. I don't want to be inhibited. Well, there are certainly things in our lives that we're inhibited from doing that would be good to do. But I dare say more often than not, we're inhibited from doing things that we shouldn't be doing. So alcohol causes people to do things that they shouldn't do. And it's kind of interesting that we expect people that consume alcohol to regulate themselves when the alcohol actually reduces their ability to do that very thing. It's just I don't know. I don't get it. But that's where we're at. Dopamine. This is going to be one of probably the main neurotransmitter mentioned in the video. This is low in Parkinson's patients. It turns muscles off, as I've already said. It also helps control mood, pleasure. It's involved in lots of different things. Let's pick on marijuana. Yeah, because boy, that's another one. That's popular. Everybody should be smoking pot, right? And so if you think of your famous pot smokers, oh yeah, we got to go Snoop Dogg here, right? Because Snoop Dogg, he's a pillar of the community. Well, what does marijuana smoking do for you? Well, it stimulates dopamine and anandamide release. So those are inhibitory neurotransmitters. It blocks glutamate, which is excitatory. You get a mild euphoria from it, but you get impairment of senses, impairment of coordination, memory, motivation, and learning. The New England Journal of Medicine says occasional use results in impaired short-term memory, impaired motor coordination, altered judgment, in high doses, paranoia and psychoses. Generally, people that use marijuana describe a diminished life satisfaction and achievement. It affects the pleasure center of the brain so that while they smoke, they're feeling good, but afterwards, they just don't feel good at all. It lowers IQ. Research says that on average, it lowers IQ 8 points. And it's particularly a problem in adolescence. That would be you folks. I know that's not always the nicest word to think of oneself, but your brain is still forming until you're 25 years old. So some of you are over 25, I know, but for the most part, it's just something you really want to stay away from because it affects brain development. And it's something that we see people increasingly addicted to. Another thing. Yeah, it's safer than cigarettes, right? No. New research says it's more damaging to the lungs than cigarette smoke is. And everybody says, well, it's got all these attributes that we don't know, and they have all these healthful benefits. Well, the truth is, we don't know what those things do, and some of them might have positive effects. Some of them might have, and we know do have, very bad effects as well. So cocaine is another one. This blocks dopamine removal from the pleasure center. So if that pathway is the pleasure center in the brain, cocaine keeps dopamine right down here in the cleft, and the person keeps feeling very pleasurable feelings, gets euphoric, self-confident, describes having an unusual clarity of thought, less desire to sleep and eat, but it is excitotoxic. And excitotoxic means that when dopamine continues to stay in the cleft here, it's going to overstimulate these receptors. Your body doesn't like that. So in response to that, it's going to pare these back. It's going to take them away. It's going to kill off some of these pathways. That's what excitotoxic means. And that means that you've got to have more cocaine to achieve the same high because there are less receptors. And it's an unending cycle. Ice, methamphetamine, again, same effect as cocaine, but lasts longer, cheaper, and depending on how it's administered, gets into the system quicker. Serotonin, very popular one, affects sleep onset. Basically, the more serotonin you have, the less sleepy you are. The less serotonin you have, the more melatonin you will have, and the sleepier you will be. And the more of an appetite you'll have. Okay. Acid or LSD binds to serotonin receptors and it distorts visual reality. People talked when I was a kid. You'd hear them describing having a bad trip or a good trip. But the problem with it's also excitotoxic, and some of those abused drugs would leave lasting effects in that out of the blue, 5, 10, 15, 20 years later, people would have a flashback, and very destructive drug. Again, it's excitotoxic as well. Endorphins and enkephalins. I talked about those when in the muscle stimulation video clip that I did for you. They're what we call natural opiates, and your body doesn't make too many of them. Is the thing. They give you what's called runner's high, a mild euphoria, and pain reduction, like after running a long way. Heroin actually mimics them, and of course, heroin is one of the drugs of choice in this day and age, unfortunately. It is also excitotoxic, but the problem with heroin is that it does what endorphins and enkephalins do to so much greater of a degree that abusers of heroin will die of basically respiratory depression. They stop breathing. They are so I don't know what you'd say. Their respiratory system is so suppressed that they don't take in enough oxygen, kick out enough CO2 anymore, and they die of respiratory failure. Sad. Again, excitotoxic. The three last lines. Withdrawal. A person going through withdrawal experiences extreme fatigue, depression, increased appetite, and you'll see some of this in the video. Tolerance, I've already alluded to that. The body makes less neurotransmitter because it's been in the cleft too long, and so the person needs more drug to get the same effect. And excitotoxicity, when there is too much drug, the body will pare back receptors and actually neurons along that pathway because there is an overstimulation of them. Okay, my intention is to get this video on Moodle. I'm hoping that goes well. I will have the study guide on Moodle as well a little later this week. I will have a brain and eyeball dissection also on Moodle a little later this week, and those should be the last items in the class. And look for the exam. I'll give you an email describing when it will be emailed, again, just like it says in the syllabus, and that will be the culmination of this class. Thank you, and have a good day.